Okay, good afternoon, colleagues, and thank you for joining this session on immunoassays, which is a continuation of yesterday's session. We'd like to thank Prof. Bongani Kambule again for assisting us by facilitating these lectures. We hope that you find these lectures quite enlightening. At the end of the lecture, you have the opportunity to ask any questions and we can have a healthy discussion around the presentation. I thank you for your attention, and the platform is now over to you, Prof.
All right, thanks. Afternoon, everyone. Thank you for joining us again. And I think today we'll just go over immunoassays. So some of the concepts that were discussed yesterday will still be applicable. And I'll just start with just providing an overview of the definitions of what we regard as immunoassays. And what we normally note is that for immunoassays, it's based on the detection or quantification of antibody and antigen complexes. So antibody and antigen complexes are also referred to as our immunocomplexes. And these are very specific. So because antibodies pick up a specific target. So if you remember the structure of an antibody, and if you have your antigen binding site, that this will be very specific to a particular antigen. And in the case of anything that we measure in the lab, the analyte can either be an antibody or antigen or any protein of particular interest. So with immunoassays, most of the immunoassays use a similar basic principle. You have an antibody that has to bind to an antigen. So usually if you're running it through a plate, if it's a 96-well plate and you're measuring changes in absorbance or fluorescence, it's also based on the complexes that form. So for instance, you have your antibody. If this is on a coated plate, the plate will be coated with an antibody. And as you inject your sample, that particular antigen that the antibody is specific for will actually bind onto that antibody that is coated on the plate. And the rest will be, as you do the wash steps, any other presence of any other protein will wash through. And what you'll be quantifying will be the amount of target antigen that's bound to your antibody. So this applies in some assays where you're also using just changes in colorimetric, and you are measuring at different absorbance levels. But you also have different types of assays where you now actually use the presence of fluorochrome. So if you remember what we talked about, what fluorochromes are, so these are proteins that can actually fluoresce when they are being excited by light. So what happens is that you have, after the initial binding, you can have the second fluorochrome, which will carry the antibody, which will carry the fluorochrome, and then we quantify the amount of fluorescence that's coming from that labeled antibody. So a similar concept applies to newer types of assays, which are known as bead immunoassays. And I think later on in the presentation, I'll just briefly go through the newer type of bead-based immunoassays, right? In older assays, we had the radioimmunoassays. So using a radioactive isotope, and we quantifying our antigen based on radioactivity. You also have an enzyme immunoassay. This is the commonly used ELISA method. There are different types of ELISAs, so we'll also just briefly go through those. And there's the fluoroimmunoassay. And lastly, I've included one of the newer type of assays, which are known as bead-based immunoassays. So with the radioimmunoassay, there's a use of a radioisotope, which is then attached to the antigen of interest, and it's bound to its complementary antibody. The antibody bound antigen is then separated from the free antigen, from the free supernatant, and we use the quantification, which is based on radioactivity. So if you look at the diagram that are provided in the slide, that the following, these are the steps. Usually you use a primary antibody, and there's also a secondary antibody, which will provide the precipitate, or will form the particular complexes that you're measuring. So we add a fixed amount of antibodies, and then we add the radioactive antigen, which will be displaced from the antibody molecules. And the final steps will be that the antigen-bound antigen is then separated from the free antigen, and in the supernatant fluid, and the radioactivity is then measured, which then will be used as quantitative measure of the presence of a particular analyte. Right. The most common type of
Sorry, may I ask a question? It's a small group, so I took the liberty of interrupting you, if you don't mind. Or would you rather we hold our questions to the end?
I think you can ask. It's no problem.
Okay. No, the radio immunoassay, it confuses me. If you could just go through it one more time slowly, please. If you have enough time to complete your presentation, I don't want to lengthen the presentation.
Okay.
So you said that you add in the antigen that is already labeled with your with the radioisotope, is it?
So so you have a radioactive antigen to the you're adding a radioactive antigen to the mixture. So you've got the antigen that you are looking. So you add a labeled amount of unlabeled antigen into the mixture, which then these compete and bind to the antibody. So I think I'm not sure what exactly you are asking. Are you asking the antigen or the isotope?
No. So you are actually adding in what you want to measure, but whatever you're adding in what you want to measure is labeled. So is it a competitive assay? Is it?
Yes, it's a competitive assay. So it's a competitive assay. It works on the principle that if you're adding so when you look at the blue molecules there, so that will be your sort of your
labeled antigen.
Yeah, that will be your labeled antigen, and that will then cause the movement of whatever is bound. It will be displaced. So your radioactive antigen will be then displaced from the molecule by your unlabeled antigen. I hope that's
okay. And once it's beginning to make some sense. So is the antibody, if you measure in the antigen, are you adding in antibody and label antigen?
Yeah. So you are adding, firstly, you have to add a radioactive antigen. That will be the one that will be displaced from the antibody. And then you're going to have finally binding of your antigen. So more of a competitive type of binding, as you were mentioning. And then
when you, okay, and when you measure in, so the lower the measurement, the more likely you have sample, more likely you have the antigen of interest.
Correctly. So that's an inverse relationship. That's correct.
Okay, but now how are you washing away the labeled antigen that is now unbound? Because it's all in solution, isn't it? Your antibody, label antigen, and the analyte of interest that you're measuring are all in solution.
Yeah.
So if you wash it, you will remove everything.
Yeah. So if you wash it, you will remove anything. But there are steps. I just didn't provide the protocol for this, but there are wash steps where you can actually dry off. So because a precipitate, the one that is bound, what you're doing is actually then using off the supernatant of that. And you actually, so if you look at step 6, so step 6, we're actually looking for antibody which is separated from the free antigen. It's found in the supernatant fluid. And that is where the radioactivity is found. So remember, if there'll be a precipitate that forms when you add the secondary antibody. So because it's a precipitate, you will not have it in the supernatant. It's actually going to settle.
So the antigen, if it is labeled or unlabeled, but if it's bound to the antibody, will settle down.
Yeah, that's after adding the secondary antibody. So you have the secondary antibody that you're going to form. So this is the precipitating antibody. And what you're looking at is the the free antigen in the supernatant fluid. So that will be almost an inverse relationship to what you're actually measuring as in the antigen.
Okay. It makes a lot more sense to me now. Thank you so much. I appreciate it.
No
so your precipitating antibody, does it have a particular property, or is it basically based on the weight of the molecule that is finally created?
Yeah, so I think it depends on the test, but there are different types of secondary antibodies, and unfortunately, I didn't provide you with the SOPs here, but
No, we just want broad principles. I'm never going to do this test in my life, but just it's nice to have a good understanding.
Yeah. No, so the secondary is just the one that causes the precipitate form. And I'm not sure if it's based on molecular weight or not, but it just causes the precipitate. And it allows that you're measuring the antigen that is now on the supernatant.
And then the radioactivity, is it measured in the supernatant or is it measured in the precipitate?
Yeah, so you're taking the supernatant, so the supernatant fluid. So you look at the free antigen and the supernatant fluid, and that's where the radioactivity is actually measured. So you're still going to have, remember, the radioactive epitope is the one which was either in iodine, and an isotope usually it's like the 125, which is then added.
Right.
So that's the one that is actually radioactive. So the amount of that free analyte that you're actually having, that's the one that gives you, if it's more that's been picked up, it's like an inverse type of relationship between what's in the sample and what you were quantifying.
So if you have a high radioactivity in your supernatant, does that mean that you have got a lot of whatever you're looking for is high or low?
It's inverse, so it will be low. So it's an inverse
Low.
relationship. Yeah.
Okay, all right. Thank you very much. I'm sorry I had to take you back to it.
No problem. Yeah, so these are one of the older essays. I personally haven't done this type of essay, but I think the principles which apply is similar, like you're saying, with a competitive assay, where you are adding an antigen to compete for antibody binding, and the more displacement you get, it gives you a sort of like a proportional or inversely proportional quantification of what's in the sample. Yeah, okay. So, oops, how do I move forward? Right. So one of the most common are the enzyme-linked immunoabsorbent assays, where you actually have antibodies that are labeled with fluorescent probes. So they emit light when excited by light, and you usually run this through a plate reader at different wavelengths. And you have an antibody-antigen complex that is formed, and the intensity of that fluorescence can either be quantified. So you've got different types of ELISAs. We've got the direct, we've got indirect, sandwich, and competitive. And I think this is what most people use. And again, it's based on the principles that if you're running a direct ELISA, you would have your sample, you have a plate. So it's either a plate where you have your sample of interest and you're just adding your primary antibody. So some are really coated, you have coated, and some you have plates that are not coated. In the olden days, you would do the coating of your cells. So you'd put in any protein, usually we were using milk, to ensure that your antigen actually attaches to the surface of your well. And what happens is that you're adding an enzyme and substrate, and you're adding an antibody that will then pick up that particular antigen that you of interest, and then you quantify. That's in a direct ELISA. In an indirect, you need a secondary antibody to pick up the primary antibody that will be bound to the target antigen, and your substrate that will generate the fluorescence that you'll actually be measuring. Right. On the third, you have the capture antibody. So with a capture antibody, we actually have an antibody that's going to cross-link. So you have an antigen that's going to cause a cross-linking. You get a primary antibody. If the plate is coated, you're adding there's an antigen, and then there'll be a secondary antibody that binds to that antigen before you actually picking up any substrate formation. You also, in your competitive ELISA, the principle is that you're just adding an inhibitor antigen, and that will cause competition or binding for the target region or antibody binding sites on your monoclonal antibodies, and you quantify that. So if you've got less of your or more of your inhibitor binding, it just tells you have less of your target antigen. So it's an inverse relationship. So if you've got less of your inhibitor binding on that, there's more binding sites for an inhibitor, that means there's less antigen in your sample, and vice versa. So it applies on the negative relationship between the signal that's quantified, that's measured, versus your target antigen, right? So there are advantages and disadvantages of different types of ELISAs. The direct ELISA, it's rapid, but it has low sensitivity. And when you talk about indirect, it has a higher sensitivity, and there's also a risk of cross-reactivity between the secondary antibody that you'll be added. So we also have a sandwich ELISA. And a sandwich ELISA, there's minimal sample purification that's needed. And it might be time consuming because of the laborious test steps that you might be doing in the lab. And you also have a competitive ELISA, the one that I've just mentioned, and it uses small antigens and there's low variability in that type of an assay, right? So another type of assay, which is one of the most commonly used assays nowadays, as known as the bead-based immunoassays. So the bead-based immunoassays, these are similar to what your Luminex platforms are using and what cytometers are using to measure analytes. And I think yesterday we had a brief discussion on just how you can actually quantify 13 different cytokines instead of doing what you would do in your normal ELISA, where you have just picking up 1 or 2 analytes using the same kit. Is that in bead-based immunoassays, you can actually design a plex and you can pick up more than 10 cytokines at a time. So, or analytes at a time. So how these work is that you are adding beads. So you have a sample. The sample is either serum, plasma, or any fluid that will contain your analyte. And what you are adding is beads. So these are latex beads and they are coated. So they are coated with a particular antibody. So when that sample or that antigen in the sample will bind to a particular antibody, that bead would then pick up a lot of these antigens, depending on how many beads antibodies are coated on that bead. And by adding the fluorochrome, so the streptavidin PE, that is the fluorochrome that you then add. There's a step of adding the fluorochrome. And what it does is that it will then bind to your detection antibody. So you have the first bead, the bead is coated with an antibody. So for instance, if this was interleukin-1, it will pick up all interleukin-1 molecules on the sample. Then I add my biotilated detection antibody, which will be conjugated with the fluorochrome. And this is only going to bind on complexes that are formed around the bead. And that is then quantified using flow cytometry. So flow cytometry or any type of a laser platform. So what this does is that the intensity that you're seeing here will be an intensity coming from the bound bead, and that is quantified and given into a standard curve that you will generate by standards, and that gives you an estimate of a concentration. So most of these immunoassays, which I didn't mention, function in a sense that you are going to have a standard, and the standard tells you what is the concentration of a particular absorbance level or intensity level, and it gives you in a concentration of the antigen you are looking at, right? So if you look at the approach for these bead complexes, is that for a bead complex, you have either any molecule, it can be hormones, it can be cytokines, it can be chemokines, but these are not in whole blood. So these are either in plasma, serum, or fluid. And then you have, instead of immunocomplex forming, you actually have a bead complex forming. So the bead is what captures all the type of molecules that you want to measure in a particular sample. And these are then on the surface. So you'll see that if this was an antibody coated to pick up adiponectin, if it was a bead coated to pick up adiponectin, you would have any molecule that is recognized as adiponectin binding onto the bead. And then we would add our secondary detection antibody, which will then form a complex. And that detection antibody is where we actually have our streptavidin PE. So the PE is the fluorochrome. So any complex that the bead, any complex that forms on the bead is what we'll then quantify. And we measure these as mean fluorescence intensity. So what that means is that the more of these complexes that form, the higher the concentration of a particular analyte. The less of these complexes form, you're going to have less binding or less forming of the bead complexes. And that tells us that there's a lower level of that particular analyte that we're looking for, right? So in the diagram, you'll see that you have quantification. So the quantification in each bead. So you have a bead population. This is done simultaneously on each sample. So you can have up until 17 cytokines that are just measured by 3 different beads. So these beads can also have multiple type of antibodies. So one bead can pick up more than one analyte, and that can be also quantified based on the MFI levels. So for the MFI levels, we then move on and quantify what type of analyte we're looking for. So there are predefined panels, and this is just an example. This is from BioLegend, but you do have others from different other suppliers. And what you have are already predefined panels. So you have a malignancy panel, you have a thrombosis panel, you have an antivirus response panel, you also have a T-cell response panel. And these have particular set of mixed mesh cytokines, the chemokines, which the beads will pick up, and they then be quantified into picograms per microliter, or depending whatever sample concentration you want. Right. So I just also wanted to show some of the human panels. So the human T-cell cytokine panel, again, the advantages of the bead assays compared to the other immunoassays, where you're using a huge amount of serum or huge amount of sample, is that with these, you don't really need to run in triplicates or duplicates because you're measuring or enumerating a certain amount of beads. So you're running 2,000 beads, and the amount of variation that you get from this is quite very minimal. So you can measure a lot of cytokines using just that one bead pump complex, and it is non-variable, right? So I think just in summary, although I didn't want to go into the protocols that we used, I think although this was just a brief overview of the concepts, and I wanted to summarize this by just repeating that with immunoassays, we talk about immunocomplexes as an indicator of the presence of an analyte. And then these could be used to determine the presence of toxins, allergens, enzymes, and drugs in samples. So these are used in different platforms. They can be used to pick up antigens and antibodies, and they can also be used to pick up certain presence of substances or even the presence of particular enzymes, right? And I think for this talk, I'd like to end here and open it up probably for any discussion. And also factoring in that I think in preparing this, I didn't want to go into detail with the technical nits and bits of the SOPs, but if anyone wants to discuss those, I'm willing to share.
Okay, thanks, Prof. Kambule. It's a very interesting presentation. The floor is now open for any questions and discussions.
Hi, Natalie. May I ask a couple questions?
Sure, Prof. Musa, go ahead. Thank you.
Thank you so much. Thank you, Dr. Kambule. I really appreciate it. You know, you are describing these assays that have always bugged me, and I understood most of how they work, but there are a few nuances that I just couldn't grasp. Would it be possible for us to go back to the bead assay a couple slides earlier, if you don't mind? You see, it sounds like it's very much like a sandwich assay, and your solid phase is actually the bead. Would that be a decent summary of what you explained?
Perfect. Yes.
All right. So my issue is that when you add in the antibody, the biotinylated antibody, and then you add in the streptavidin with whatever detection molecule you have, how does it create the specificity for the different analytes? Because the biotinylated antibody is common to all the analytes, because the analytes are all bound to antibodies.
Yeah.
Am I right? Because
yeah.
So if you can just explain that, I can't seem to wrap my mind around that. I'm sure there's a good explanation, but I am having difficulty understanding that.
Yeah. So that's a very difficult one for me too. But from what I understand about this, that so you have the particular detection antibody, and you're right in saying that it appears that it binds to the antigen like a sandwich, but it will be questionable for it to have the same specificity as the bead, which is actually has the particular antigen and antibody specific to the antigen that it's picking it up. Unfortunately, I can't give you a clear answer on how these work, because some of these products we really don't know what the technology that's used for that particular biointerlated detection antibody is, but from what I know is that the detection antibody that you add on almost all of these are similar, and the only difference is that it actually has the fluorochrome that's picking up, but it somehow does detect where there's a complex. So I think the major principle is that the capture antibody is the one that picks up your analyte. So the detection antibody.
Do you think the detection antibody and the capture antibody are the same?
No.
You see, I'm just wondering, because if the detection antibody and the capture antibody are the same, then that would create specificity, wouldn't it?
Yeah. But
your fluorescence will have to be different, because your flow cytometry differentiates molecules based on different fluorescence. Am I correct?
Yeah. So with the bead array, it differs. We don't. So we have two channels with the bead arrays. So we have a reporter channel. So if you look at these one down here, you have a red channel. So the red channel is a detector channel. So we identify the beads, and we have a quantification channel. So that's PE. So for these type of assays, we're just using two. We don't use fluorochromes to work out different antigens. So you just use two fluorochromes. It's red. It's usually APC that picks up the antigens, and all your antibodies, all your cytokines are measured on the same channel. So it's quite different from your standard
right.
cytometry. Yeah.
So I'm just wondering, how does the machine know that it's picking up different analytes if it's using the same channel? You understand my confusion?
Yeah, I understand. So what it does is that it's just that, again, these beads are labeled. So when you add the beads, you're adding like 6 or 8 beads. So yeah, I don't know if you see my cursor, there's a thing that's written
yes.
bead A4, A5, B4, and B7, right? So these beads differ. What you do is that you create a gate. So you will know that they will tell you that the size of bead A4 will be high up on the red spectrum, right? So that's the bead population. So you are able to pick up, if the red bead is for adiponectin, anything that has formed the complex on the red bead is adiponectin. Anything that's formed a complex on the sort of like pinkish or a peach bead is actually leptin. So that's how it works. You're actually gating on the bead, and the bead will have specific antibodies for an analyte. Yeah. So you can have.
So how are the beads different? And now I'm understanding what you are saying. So somehow or the other, the beads are different enough so that they can be picked up differently on that scatter sheet.
Yes.
Scatter, whatever you call it.
Yeah. So if you look at the example of this panel, this human panel, right? You can see that there's like bead A4 to bead B9. So these are all different beads. They have a different capture antibody, which is the one that is specific for the analyte. So the capture antibody will be a capture antibody for adiponectin, interferon, and so on. But the streptavidin comes in as one reagent. It's the same for everything. It just carries the streptavidin and the biotilated antibody. So I think what this happens, this secondary, if we call it secondary in the case of the sandwich, this biotilated detection antibody somehow picks up where there's a complex, where there's an antibody-antigen complex, and binds on it. But when we then run it through the cytometer, we are able to say that peach population is actually bead A4. So anything that comes from this, on this histogram, is the levels of adiponectin.
Okay.
So anything that comes from these bead population, if it's an interleukin 6 bead, it's actually interleukin 6. So it works.
So there's something different about the beads that allows them to appear differently on your, what do you call it? Do you call it a scatter plot? I'm used to flow cytometry and when they differentiate in cells. What do you call these different regions within your plot?
Yeah, so you're correct. So this regions, like if you look at this, this will be, you create a scatter plot. So you create X and Y scatter plot. So you're just putting 2 different filters. So if you've got FITC versus PE as the standard, you are going to get the separation based on the fluorescence. So the beads fluoresce at different intensities. So if you look at the MFI on the red, you'll see that the peach is quite higher than that red population. So the beads themselves have different fluorescent properties that you're picking up on the reporter. So you've got the reporter channel. So, but this doesn't interfere with the quantification of your cytokines, because the quantification is done on a different fluorescent filter.
No, thank you very much. I appreciate it.
No, it's no problem.
Now, often when you don't know something, it's reassuring when you know other people don't know it. It makes you feel better about your ignorance.
Yeah.
So,
Yeah.
so sorry if
No problem. Some of these, like the technology of how it picks up the complexes, unfortunately, we're not privy to that. I mean, I've used this kit several times. What I'm saying is that you hardly get the molecular information of whether this is looking at binding on the antigen or just randomly binding to all antigens. And if so, how does it detect? Is it a matter of affinity? Because there's a lot of wash steps in this protocol. So is it a matter of affinity or not? That's not clear.
Yeah. I have another one or two questions, but I'll allow anyone else to ask their question and then I can come back to it. Thank you. Thanks, Natalie. Let others have a chance. I don't want to hold.
Prof, I don't think anyone's asking. You can go ahead. It's fine. If our colleagues are but cannot unmute their mic, sorry, they're free to even put their comments or questions on the chat and I will read it to Prof. Kambole afterwards. So go ahead, Prof. Musa.
Please call me Yunus, Natalie.
Okay, sure.
Can we go back a couple slides? You know, when you looked at the direct fluorescent testing, I've seen that in the context of, for example, when somebody has got pneumocystis pneumonia, for example, then a tissue sample is given, and then you use an antibody that is directed against pneumocystis that is labeled, and you can see it on the tissue as fluorescence. But that would probably be considered a direct fluorescent antigen test. But here you are talking about a direct ELISA, and I'm just wondering, how would you do that to measure and analyze? I just couldn't understand it. Is that something that that is done? And if it is done, do you have a better sense? Maybe you can explain it to me.
Yeah, so direct ELISAs are the most commonly done. So what we do is that if you take your particular sample, we add the analyte, so whether it's serum or we normally do this for even, can't even think of an assay now. But what you do is that in a direct, you just need to make sure that it's protein coated. So usually, so that if you're adding your serum and you're doing the wash steps, you remember there are two ways where you start by the coated and you're actually using an antibody coated. The others are protein coated ELISAs. So when you add your antigens and you add your antibody, you're able to pick up the presence of that particular antibody. So some don't do the several wash steps, but these is just based on you directly adding the antibody and picking up a particular target of interest. So I'm not sure whether you're questioning whether in tissue you wouldn't have issues with...
You see, in tissue, you have automatically created a solid phase because your tissue forms the matrix in which your antigen is embedded and you're actually looking for that antigen. So it's very understandable doing a DFA, which is a direct fluorescent antibody test. But when you are looking for an analyte, for example, if that green ball that you see on the plate on your slide is an antigen, and if you add in an antibody, if you are detecting the antigen, well, it's already on your slide because it's there. You know it's there. You can't be measuring that. And if you can't be measuring the antibody, because for you to see the antibody, it needs to be fluorescent labeled. So you can't be measuring it. It's something that you would have synthesized and put it in. So I'm wondering, how do you use direct ELISAs to measure anything?
Yeah, so
am I confused?
I sort of get you, but not clearly. But I think that, so in a direct ELISA, so I think maybe the first part is that the plate is not just plastic plate without any type of coating, right? So you are actually binding, you actually are getting coated plates. So you can buy coated plates and you can also have biotin binding plates, right? So these are also coated. And this will ensure that either for the biotin, you're actually having some form of binding that occurs between your antibodies and every target that you're adding. So in this case, you are taking a serum sample that will have the target antigen. You're loading it onto your plate. And then you are adding all your other antibodies and your substrate to pick up if that antibody does bind after a wash step, whatever still remains in that particular plate that's stuck on the bottom of the well and also has the particular antibody with the substrate on it, with the target antibody. And as you're running it and you're picking up whether it's in a case of absorption, or if it's a case of fluorescence, then that is quantified as directly the presence of an antigen.
So in that case, would you be measuring the antigen?
Yes, you're measuring the presence
you
of an antigen. Yeah.
So that means your antigen, when you add your plasma sample to this particular slide or whatever plate, it would bind that antigen, and then
Yes.
you're using your antibody to detect whether the antigen is there.
Is there? Yes, correct.
I get
That's
you.
correct.
I get
That's
you.
correct. That's correct. Yeah. So I think the ones that are a bit tricky when it comes to using in competitive ELISAs. So with competitive ELISAs, it's a bit tricky because you're also adding an inhibitor antigen. And I think it's an interpretation of inverse in this case. So I would have I say my sample has the presence of antigen A, and if antigen A is present, then it's an infection or whatever it is. But I also add an inhibitor antigen that will compete for the binding of that unknown antigen that's in my sample. And after, if I wash, and then I look at the amount of unbound inhibitor antigen, if there's more bound inhibitor antigen, then there's less of the target analyte or target antigen. So these are the ones that are often a bit difficult because they also use the similar approach of just picking up an antigen, but you also are adding an inhibitor. So they are appropriate for different types of assays. But I think that the take home is that most of the time in the lab, people lose the antigen because some of the plates are not coated and you have to do the coating yourself. So if you just load a sample and you add all the other antibodies and you perform a wash, because nothing has actually bound to the surface of the well, you're actually getting no read at the end.
I get you. I have one other question if nobody else has any questions. You know, in medicine, we often talk about the prozone phenomenon. Are you familiar with it? I don't know if you really do it in the lab. You're not familiar. The prozone effect is where when you have too much of what you want to measure, the analyte, you can actually get a negative test. So in other words, you would get a false negative, but if you dilute the sample serially, it then becomes positive. So if you have a very high suspicion that the patient has a particular disease, and then you will alert the lab that there is a possibility that you might get a false negative because of the prozone effect. So they should dilute the sample to continuously looking for the analyte, and it will become positive.
Yeah.
So you understand what I'm saying?
Yes, yes.
Now, I have a difficulty in wrapping my mind about why the prozone effect occurs. And when I did a Google search, they talk about, an antigen-antibody complex needs to be of a particular ratio for you to see a precipitate, because when the antigen-antibody bind to each other, they form a lattice and they precipitate out of solution. And that you need a specific proportion of antigen-antibody. And if you have an excess of either, you won't form a precipitate. But as you dilute the sample, you reach a point where there is the appropriate concentration of both your antigen that you might be measuring and the antibody that you are using to detect the antigen. And therefore they will precipitate out, and therefore you will note it. That's what they explain. And I just don't understand it. So I thought maybe if you understood it, you could explain it.
Yeah, I think just dealing with all the immunoassays, which is also similar to the bead-based or even flow cytometry-based detection, is that if you've got an excess of, for instance, in this case, if you're detecting patient and the presence of an antibody, and you're using indirect detection methods, if it's too concentrated, you also have this non-specific binding in the sense that would happen. But what I don't get is that you're not getting a read. So usually all these immunoassays have a standard and they have a detection limit. So when you set them up in the lab, you run a standard curve which has a lower detection limit and an output detection limit. So I don't know if you've seen before a curve, a standard curve, or what the standard curve looks like. So in most cases, if the patient sample or your test sample is too concentrated, it will not fall within the detection bounds. So you may not see any when you try and actually work back the concentration. And I'm talking about quantitative assays here. That is when we would do the serial dilutions. And that is more of just ensuring that our sample falls between the standards that we can actually use to extrapolate the quantitative values. But on the side of the letters, I'm really not sure how that works. I haven't encountered that in any of the essays that I've done before. So probably maybe
no, that's fine. That's fine. I just that this is something that we often in clinical medicine, people talk about it and people make you aware that that could happen. You don't see it often, but it's something we are aware of. So I thought maybe you could, being in the lab and understanding all of these dynamics, you might be able to help me understanding it.
All right. Oh, thanks for the question, Prof.
You're welcome. You're welcome.
I see there's a question by Brenda. So Brenda wants to know if there is going to be a planned practical session to supplement the theory that is now provided. So when we started the series of lectures, we didn't plan on any techniques course, but I'm sure, Bongani, what do you think that probably next year, based on your availability, we could run a techniques course to supplement the theory that has now been taught. Yeah.
No, thanks, Brenda, for those. Yeah, and I think that is one of the things I've been trying to get going, is because we have all these high-tech instruments that most people are not aware of in different disciplines, that are situated in different areas or different labs within the university, which are instruments that should be used for training. So yeah, it's something that I would like to be involved in, and I think it could be good to build up on that, and also just to ensure that if there's anyone else from the clinical side who actually wants to have some time and actually see how some of these essays are set up, and maybe also understanding some of the technical nitty-gritties of it, then it's something that it's worth setting up and worth doing. We do have several flow cytometers. We have a sorter, which I would really like to motivate anyone on the call to also start using. And with the sorter, you're picking up specific cells, you can isolate these cells, and you can do a lot of things on a cell-specific thing. So if you get viral infections, you can pick up the infected cells based on the antigens that you're looking at, isolate just the cell instead of reporting on an average number of cells that are showing an antigen. And you can actually run particular assays to look at a specific isolated cell. So we also do magnetic bead sorting. So in Westville, in the lab in physiology, we've set up, it's a magnetic immunoassay. So you're just taking samples, the antibodies in this case are coated with a magnet. So if I'm looking for an antibody that's going to detect a viral particle, the antibody itself is coated with an antibody, with a magnet. You run it through a magnetic field and it's able to pick up all the antibodies that are bound, and you can actually quantify those, and it gives you, so these are all high-tech techniques that are just more than an ELISA, and I think bridging people moving from the theoretical part into the practicalities of it is something that I would also really encourage that we actually do if we can next year.
Thanks, Bongani. I think definitely we'd look at running such a course and opening it up to our colleagues within the college, students as well as staff. I'm sure it'll be great. Thanks, Brenda, for bringing that to our attention, and that's something that we could look at developing for early 2022. Are there any more questions or comments before we close the session?
I'd just like to say thank you so much for these sessions. I think we need to encourage clinicians to join these sessions, because if they understand laboratory tests better, they can understand what can go wrong in a test and therefore query results that just don't make sense to them. It's not about just doing the test and being able to do the test. That's not important for the clinician. But I think clinicians need to understand how these tests are done and how things can go wrong in a test that will inform them better to be more critical of results that they get. But thank you so much for arranging these. I appreciate it.
Thanks.
By the way, I asked my department to join, and it appears nobody has joined.
But thank you for joining, and thank you for the very nice discussions and the excellent questions. It really kept the momentum going when it came to this talk. Thank you for that. So colleagues, thank you so much for attending the session. This is our last session for the year. we would resume next year with more interesting talks. I would have all of these links to the various presentations uploaded on the Clinical Medicine Lab webpage. So you can share them with your colleagues and watch them for your own interest if you've missed out any sessions. We're taking all your comments into consideration, and we hope to see you next year for our techniques course. Thank you and have a good afternoon. And sorry about the load shedding, but despite that, have a good weekend as well.
Thank you so much. Bye, everybody.
Bye.
All right.